sailor fell asleep during his shift. This morning we found wrapped around his ankle up into the sleeping bag. It didn't even wake him. The rate of growth is... Rodriguez says he's finished the translation. Apparently the carvings along the eastern wall show human forms inside something. We thought they were purely symbolic, but the proportions, the anatomical detail, the clearing is shrinking. We're activating containment protocols. But I don't think we can stay here. I keep thinking, what if they're not meant to keep things out? What if...
That was the final recording received from Dr. Lin Chen, a biochemist with 14 years of field experience. She published papers on extremophile bacteria. She had a reputation for methodical, careful work, which made what happened at that temple all the more disturbing. The official report blamed a flash flood, early storm season. This explanation would have satisfied me if the meteorological data hadn't shown only moderate rainfall for the entire month, if the equipment hadn't been carefully packed and powered down, and if Chen's final transmission hadn't mentioned containment protocols. And so it was that my team and I were dispatched, sent to investigate the disappearance of Chen's entire archaeological team. In my years of study, I've encountered organisms that push the boundaries of known science. Creatures that challenge our taxonomic classifications, even ones that require entirely new theoretical frameworks. But what we found at that temple, well, it was certainly no creature. We landed at a regional airstrip 5 days after the company lost contact with Chen's team. The journey to the site took another 2 days, first by Land Rover through progressively deteriorating roads, then on foot when the jungle reclaimed even those. Our guide was a man who'd worked with archaeological teams for 2 decades. He stopped at a creek bed 6 kilometers from the temple and refused to go further. The locals considered the area forbidden, he said, and had done so for generations. When we asked him for specifics, he only repeated the same phrase.
Tierra hambrienta.
Of course, I'd heard similar warnings at other sites. Usually they were cultural memories of real but mundane dangers, unstable terrain, disease vectors, predator territories. But the fear in our guide's expression was deeper. Over the years, I've come to believe that there are places where our understanding grows thin. Unscientific, perhaps, but standing at the edge of that green wall, listening to the jungle's complete silence, well, I couldn't entirely dismiss the observation. The hike to Chen's camp should have taken 3 hours. It took 5. The jungle resisted our progress, and by the time we glimpsed the temple's uncovered stone crown rising above the canopy, it was late afternoon. Chen's base camp spread across a cleared area of what had probably been around 50 meters from the temple entrance, but was already being reclaimed by plant growth. At first glance, nothing seemed truly amiss. Tents stood in neat rows, solar panels were angled toward breaks in the canopy. The kitchen area remained organized, supplies stacked in waterproof crates marked with inventory numbers, and in the central tent we even discovered Chen's field notes open on her desk. Her last entry dated the morning of her final transmission. In short, there were no signs of distress or panic, and yet the only signs of life here were the plants, pressing into the clearing from all sides as though they were eavesdropping on our hushed conversations. And something felt off. It took some time to realize what it was. Other than the whisper of wind through the trees, this area was completely silent. No bird calls, no droning insects or distant whoops of primates. Only once did something break that stillness, almost like a ship's timbers straining, but wet, organic. It seemed to come from everywhere and nowhere, lasting maybe 5 seconds before silence reclaimed the space. We made note of it and moved on. We had only 5 hours of daylight remaining, and I made the decision to begin our survey immediately, beginning with the temple that loomed before us. Its entrance a carved mouth in the hillside that seemed to swallow light. The corridor of that entrance sloped downward, and our flashlights could carve only weak cones into its depths. It felt less like a hallway and more like a throat, the air thick, humid, digestive. We went in hoping to find answers, but for now at least, all we'd find were more questions. It was hard to tell just how old the temple was. It was made of stone blocks fitted without mortar, worn smooth by centuries but showing no signs of structural weakness. It was strange, but the entire architecture seemed to be designed backward. Defensive positions faced inward. What were undoubtedly channels for drainage ran uphill, and about 20 meters into the descent, we began to see carvings on the walls. Now, typical Mayan iconography emphasizes human figures and celestial bodies, but in these, center stage belonged to something else. Botanical motifs, certainly, but rendered with unusually precise anatomical detail. There were cross-sections of what appeared to be seed pods, and root systems mapped like circulatory diagrams, and those human figures throughout. Flashes rang out through the darkness as Allison documented the glyphs, each one like the blow of a hammer in this enclosed space. I noticed that one of these carvings depicted human forms arranged inside what appeared to be pods or chambers. The detail was extraordinary. That night, we established our own camp at what we judged a safe distance from both the temple and Chen's site. The protocol was standard: establish a perimeter, set a watch rotation. I woke for my shift at 0300. The air was cooler now, misty, our own clearing barely illuminated by moonlight, and that pervasive sense of complete silence. It was then that I noticed, or thought I noticed, the edge of the clearing seemed closer to my tent than it had before. At the time, I thought that ridiculous, a trick of the pale light and fog, or a misremembering. We found more of Chen's field notes the next morning, sealed in waterproof containers near the main tent. Her early entries clearly conveyed the excitement of discovering an unknown cultural subset. But by day 8, her tone had changed.
Rodriguez insists the root systems are expanding. I didn't believe it at first, but we've taken photographs every day, and we've confirmed it. We calculate now a growth rate of roughly 2.3 centimeters per hour during peak periods, which is impossible.
Day 12.
Miller woke up last night yelling. The growth had reached the interior of his tent and around his sleeping bag. We cut it away easily enough, but regrowth reached the same position within 4 hours. I also noted unusual swellings at the distal tips. They don't seem to be consistent with leaf or bud formation. They could be reproductive structures.
Her final entry, scrawled in deteriorating handwriting.
The clearing is shrinking. I measured it this morning. 8 meters lost on the north side alone. How did we not notice before? The perimeter is now at 12 meters. If this rate continues, I estimate complete enclosure within 72 hours. I think we'll have to attempt extraction at dawn.
I closed the journal as the morning sun struggled through the canopy. Around us, the jungle maintained its unnatural quiet. My pulse quickened as I thought back to those early morning hours. Had our perimeter always been this small? When we'd arrived 3 days ago, hadn't there been more space? Marcus was already measuring. His expression told me what I feared. Somehow we'd lost 2 meters. Uniformly, on all sides. By our 4th day, Marcus had begun to track the encroachment patterns, even creating a map of the growth. Hoping to learn more, we determined to follow a particularly dense growth zone that led deeper into the jungle east of the temple. The path wound between buttress roots and through dense undergrowth, and 20 meters in, the forest floor changed, became softer, spongier. We found the bones in a natural depression perhaps 3 meters across, like a bed of interwoven roots. Animal remains first. Small mammals, their skeleton intact but strangely eroded. The bone's surface showed what appeared to be chemical weathering. I had seen similar erosion patterns before, but in a lab. A kind of pitting that occurs when bone tissue is suspended in proteolytic solution. Allison once again began to document the scene, and I took a careless step toward the depression. The response was instant. The underlying network of plants, what I'd taken for thickened roots, contracted inward with a wet snap, enclosing the area just in front of my boot faster than a blink. It was like a Venus flytrap, but as we'd come to discover, the trigger zones extended for meters. My one misplaced footfall had activated a single contraction, but there were certain to be more trigger zones, perhaps all around us. To my discomfort, I realized that we were standing in a biological minefield. And it was then that Allison spotted the human remains. A skull partially buried, showing those same patterns of erosion, patches of scalp still clinging to degraded bone. We photographed everything, measured the depression, took samples of the soil and of the plant itself. But as we documented the scene, I became aware that the growth around the depression had shifted since our arrival. We retreated carefully, retracing our steps as best as we could. And that sound, that wet creaking, followed us all the way back to camp. When we arrived, our clearing had become even smaller. Large tendrils of vine now extended from the jungle's edge, snaking along the ground toward our tents like fingers feeling blindly in the darkness. Traps were one thing, Though we saw no perceptible movement, I couldn't shake the feeling that now we were being hunted. When confronted with horror, the scientific mind seeks refuge in methodology. And so we were all more than willing to spend the next 6 hours establishing a field laboratory, processing samples, and applying the framework of botanical science to something that by all accounts defied easy classification. Still, for the purposes of study, I have called this species Pseudoyataveo sanguinaria. Now, it goes without saying that plants, by their nature, make poor predators. Rooted in place, they rely on prey coming to them. The Venus flytrap achieves this with sweet nectar, snapping shut when trigger hairs detect an insect. Pitcher plants create slippery death traps filled with digestive fluid. Sundews use adhesive tentacles, but those plants feed on insects, small frogs at most. Legends of man-eating plants, like the Madagascar tree that supposedly consumed humans, the Yatebeo with its flailing vines, have always been dismissed as colonial fantasies. But even our first tissue samples challenged all of those assumptions. The cellular architecture certainly resembled known carnivorous plants, perhaps modified from the order Ericales, but the scale was wrong. Whereas a pitcher plant's digestive glands might span millimeters, these structures extended through entire root systems. I also noted thick walled cells clustered at certain nodes, their purpose unclear. They reminded me of the pressurized cells in the seed pods of impatiens, but far more robust. The enzyme concentrations in the soil explained the bone erosion patterns I'd recognized from my laboratory work. These proteins didn't just break down tissue, they liquefied it. We observed proteases that could cleave peptide bonds not in days, but in mere hours, and phosphatases that extracted calcium from bone. Put simply, a mammalian body trapped by one of these plants would dissolve completely, every useful molecule absorbed and transported through the root system, even one as large as a human. We had learned much of this plant's digestive process, but we hadn't yet determined how exactly it trapped its prey. Our answer came from Marcus's detailed tracking of overnight growth. You see, normal vines spread through a process called circumnutation. They spiral slowly, eventually finding purchase on some higher location. I had briefly studied this in Convolvulus, watching time-lapse footage of morning glories tracing lazy circles as they grew. Sanguinaria was different. It telescopes, so to speak, from specialized nodes, much like bamboo, which can grow up to 35 inches in a single day, through cell elongation rather than division. Preformed cells simply expand by pumping in water. This organism had developed a similar mechanism, but retained flexibility. As Chen had noted, new growth could extend an astonishing 2.3 centimeters per hour, all while remaining as pliable as a vine. We tested it. A heated stone placed 10 meters from visible growth attracted rootlets within 6 hours. They grew directly toward the heat source, deviating only to avoid obstacles. Under the microscope, we observed what appeared to be modified TRPV-like channels, the proteins that let your tongue detect the heat of spicy food. But these responded to ambient temperature differences of just half a degree. In short, any warm-blooded animal that lingered, sleeping, resting, dying, would inevitably attract growth. I thought of Poya raimondi, the queen of the Andes, a plant that spends decades as a spiny, silent spike, then, in response to a precise convergence of warmth, altitude, and time, flowers once and dies. The comparison was imperfect, of course, but I couldn't shake the feeling that this organism had also been waiting. And each night we slept, we were training this organism exactly where to extend its hunting network. Of course, none of this growth would matter if Sanguinaria had no way to restrict its prey from escaping, and it most certainly does. And the trigger mechanism I discovered by accident proved to be astoundingly sophisticated. In plants, structural support is provided by turgor pressure, water pushing against cell walls. It's why lettuce wilts without water. The motor cells in Mimosa pudica, for example, use controlled turgor loss to fold leaves when touched, pumping out potassium ions so water follows by osmosis. The whole process takes about a second. In Sanguinaria, the same principle had been weaponized. When I stepped on that specialized root zone, thousands of motor cells had simultaneously opened ion channels. Water rushed out. By my estimates, the cells collapsed in approximately 120 milliseconds, faster than human reflexes. But where Mimosa's response ends there, in Sanguinaria, this created a vacuum that pulled surrounding tissue inward. Adjacent trigger zones, suddenly stressed, fired in sequence. I watched Marcus demonstrate with a branch, dragging it across a carefully selected region of the forest floor. The contractions followed the movement like a wave, each trigger activating the next. Even a running animal would face cascading collapses from all directions. The faster it fled, the more triggers it hit, and the more it struggled, the tighter the entanglement became. It was the perfect trap. But it wasn't until our 5th day that the true scope of the organism would reveal itself. Marcus had understandably been troubled by his measurements of encroachment patterns, and he convinced me with little effort to unpack the ground-penetrating radar we'd brought for mapping the temple foundations. He set up the equipment at the edge of our shrinking perimeter. In short, these scans indicated that what appeared on the surface as scattered growth zones were in fact connected by rhizomes the thickness of my arm, some extending hundreds of meters underground. We spent the next few hours mapping, carefully moving the radar in systematic transects. Each pass revealed more connections. This was not a colony of individual plants as I had assumed. It was a single organism spanning at least 4 hectares. It was in some ways like Pando, the ancient clonal aspen that covers 43 hectares of Utah. I traced the connections myself, injecting fluorescent dye at one location. Within an hour, traces had appeared at sampling points throughout our mapped area. The transport occurred through specialized vascular tissue at nearly 1 meter per minute, far faster than typical plant circulation. And when we triggered contractions at the north edge, thermal imaging showed metabolic increases at the south within 12 minutes. It appeared that the entire organism could direct resources toward active hunting zones. Throughout this process, a question had been nagging at me. Why hadn't this plant been documented before? Was it isolated to this precise location? It gradually became clear that sanguinaria requires specific conditions: limestone substrate, humidity above 80%, and stable temperatures. It was likely difficult to spread beyond this very valley, though certainly not impossible. Likely for millennia it had survived here, perfecting its strategy on whatever entered its domain. Other populations might exist in similar pockets, but this one, we were beginning to understand, was different. At some point, we became struck with our own predicament. The clearing was shrinking, that much had been clear, but now we were seeing just what that meant. And if we were to linger much longer, we realized it may not end well for us. And yet we had to know more, to understand. With a sense of urgency, we decided to return to the temple. We descended again into those carved corridors, but now with different eyes. Allison had been photographing the glyphs since our arrival, but their meaning had remained opaque. The botanical motifs dominated every surface: roots, vines, pod-like structures. We'd assumed they were some kind of agricultural metaphors, a fertility cult perhaps. But now, deeper into the tunnel than we'd been before, we saw panels that were likely even older, crude and weathered nearly smooth. They showed human figures fleeing from what appeared to be radiating lines. Later scenes depicted the same root-like patterns, but with human figures positioned differently, not fleeing anymore. They were being placed. These were instructions. The middle panels showed construction, human figures carving channels, the same channels we'd been walking through. Others carrying blocks of stone marked with a distinctive pattern. In the deepest chambers, we found where the channels terminated, circular openings in the floor worn smooth. Through gaps in the accumulated debris, we saw living root tissue. The channels hadn't been for drainage. They were feeding tubes. I believe that whoever built this temple had built their entire temple complex as a delivery system. They turned this organism into a controlled method of execution. For centuries, they'd likely fed it condemned prisoners, enemies, or perhaps volunteers. They'd maintained and managed it. When their civilization collapsed, the maintenance ended. The organism remained, its purpose unchanged. Even now, it waited at the terminus of those ancient channels for the next offering to descend. We located Chen's secondary observation post by accident. Allison's foot caught on a guy line still anchored in the undergrowth. The site was 200 meters north of the main camp, a minimal setup that told a story of growing desperation. Unlike the first camp, here things were in disarray. Scattered notebooks showed water damage and what appeared like burn marks. A field microscope sat uncovered, its lenses clouded by fungal growth. But it was Chen's final notes that gave us the most information.
The sound is constant now, from all directions. The team says they're unable to sleep. Rodriguez says it sounds like laughter. It obviously doesn't, but knowing that isn't helping. The incident with Miller was catastrophic. Rhizomes breached the tent floor overnight. We found him wrapped to the chest. In places, it looked like his skin had melted. I think that the growth followed the thermal gradient from his sleeping bag. I think direct escape through the jungle is too risky. I estimate trigger density at 15 to 20 per square meter. And running guarantees entanglement. Do not run through the growth. The faster you move, the more triggers activate.
That wet creaking. We'd been hearing it since arrival, dismissing it as settling wood or thermal expansion. But Chen's notes made us listen differently. The sound came in waves, rhythmic, almost respiratory. This was the organism's voice. Millions of cells undergoing rapid pressure changes created audible stress in the tissue, like hearing a tree grow, but compressed from years into seconds. The mechanism was simple. As water rushed into expanding cells, internal pressure could exceed 10 atmospheres. Multiply that by billions of cells extending simultaneously, and the collective groan became audible. I recalled one legend, not unlike this one in which a man-eating tree was said to make sounds of its own, ones that sounded in the local language like, "I see you." It didn't seem so fanciful now, but that tree was said to grow half a world away. Unless, well, no point in speculation. The sound intensified that night. When we walked the perimeter, waves of creaking followed our path. The organism was mobilizing, redistributing resources toward our heat signatures. The louder it grew, the faster it approached. By our 6th day, the situation had become critical. The clearing had shrunk to 8 meters across. Equipment at the periphery vanished beneath growth overnight. The sound was now constant, a wet, organic thunder that made sleep impossible. That night, Allison woke us from fitful sleep with a strangled cry. Rhizomes had breached her tent floor, likely following the CO2 plume from her breath. They'd wound around her ankle and wrist, not constricting, but growing with inexorable patience. In what was perhaps the most viscerally disturbing part of all of this, the plant tissue was warm to the touch. We weren't just standing on soil anymore. We were centered on a primed digestive system, waiting for us to stop moving long enough to be processed. And now the wet creaking had taken on a strained quality, punctuated by occasional sharp cracks, like ice beginning to fracture. By dawn, we noticed swellings emerging throughout the colony, pushing up through the soil like buried fists. By midday, these had developed into distinctive pods, their surfaces taut and segmented into precise geometric sections. We measured one of the larger specimens at about 30 centimeters across. The surface showed unusual strain patterns, minute cracks forming along the segment boundaries. I was collecting tissue samples when the first one detonated. The pop was sharp, explosive, perhaps 20 meters to our north, then nothing for several minutes. Then another crack, this time from the east. Through binoculars, I caught the aftermath of a third, fragments scattering in a radial pattern, too fast to track individual trajectories. The mechanism became clear through examination of a partially developed pod. The same hydraulic system that powered the trap contractions was building pressure within rigid-walled compartments. Inside, I found clusters of small, dense structures, vegetative propagules little more than preformed clones wrapped in protective tissue, like the bulbils some lilies use to reproduce, but increased in effectiveness through hydraulic pressure. The process was elegant in its simplicity. Water pumped in, but the lignified walls didn't expand. Something like Hira crepitans, the sandbox tree. But instead of months of passive drying creating tension, this was active pressurization. When pressure exceeded structural limits, the pod would rupture, launching its cargo of propagules in all directions. By late afternoon, the pops came every few minutes. Zones of older growth were dehissing first, waves of explosions working steadily inward toward our position. And through it all, that wet creaking continued, but strained now, like over-tightened cables. The entire organism was transforming itself into hundreds of botanical grenades. The decision was straightforward. Fire was our only option. We used our remaining fuel to create ignition points around the colony's perimeter. The dry season had left the outer growth desiccated, and flames spread quickly through tissue designed for fluid transport. The sound as it burned was unlike anything I'd encountered. That wet creaking became a sustained rush as superheated water vapor escaped from millions of cells. Pods ruptured in the heat, but the updraft carried most propagules into temperatures that destroyed them instantly. We retreated through the temple as the fire spread. Behind us, 4 hectares burned steadily, and that sound of cellular rupture gradually faded as we gained distance. By the time we reached safe ground, smoke was rising in a thick column visible for kilometers. The fire would burn through the night, consuming decades of growth in just hours. 3 days later, we returned to document the devastation. The surface destruction appeared complete. The feeding depressions were now bowls of ash, but in some places, pale shoots pushed up through the blackened soil. The rhizome network had survived at depth, likely insulated by soil that never exceeded lethal temperatures. The organism would regenerate, perhaps taking decades to reach its former extent, but it would return. We never found Dr. Chen or the rest of her team. The skull we discovered was too degraded for field identification. Perhaps they escaped through the temple as we did. Perhaps they made it to the surrounding villages. Or perhaps, well, the feeding depressions were numerous. We recorded what we could at the time. For now, I recommend continual monitoring and perhaps eventual controlled burns to prevent dispersal. Even now, somewhere in that valley, it grows, patient as stone. It has outlasted the civilization that tried to appease it. It will outlast myself. In the next decades, when new pods begin to swell, the only ones who remember are the ones who never forgot in the first place. The ground, it would seem, is still hungry. This video was heavily inspired by Scott Smith's The Ruins, and was made possible by my patrons and by viewers like you. Thank you, and remember, you matter.